Hello everyone, and welcome back to another Mineral Life seminar. And this is a happy coincidence that this seminar is also on arsenic because the last seminar was also about arsenic, and it kind of lays a nice foundation about understanding the arsenic geochemical cycle. So today with us we have Dr. Naresh Kumar, who is a junior group leader in environmental geoscience at the University of Vienna, Austria. He received his PhD from Aix-Marseille University in France, and he did his postdoctoral work at Stanford. His research focuses on mineral water organic matter interfaces and the implication on biogeochemical cycling of contaminants. He's interested in mechanistic understanding of biogeochemical processes occurring at environmental interfaces. So with that, I'm going to hand it to Naresh to talk about his recent work.
Thanks, Neha and Karim. Thank you so much for inviting. It's really an honor to talk about my work with you guys. And just before I start, between Marseille and Paris, there always been a rivalry, and I'm not part of that, just to be clear here. I like equally Paris and Marseille. Okay, so today in this talk, I'm going to talk about a little bit on arsenic partitioning at redox interfaces. And before I go to the research or experimental data, I'll just walk you through a little bit about what we do here in University of Vienna. I'm not sure how many people are aware of it. We are called EDGE, which is environmental geochemistry. D has no role alphabetically in this story, but it just fit well here because other departments were named on the shapes. One is dome and one is cube. So we found that easy to use that one. Our lab, we mostly focus on redox interfaces, environmental interfaces, where we look at different mechanistic things. Most focus of our work is, or let's say core of our work we do here is redox interfaces. And within that redox interfaces, we do different projects, and it's not such a big group. We have 5 PhD students, 2 postdoc, and 4 master students right now, so pretty small. And in COVID times, it's pretty good size to keep things running. Coming back to our redox interfaces, we look at contaminant mobility. One aspect of our work is contaminant mobility, how these systems impact that. And within that, we look at arsenic, uranium, chromium, mercury. That's right now projects we have on these contaminants. We also look at nutrient availability and how that is impacted by redox interfaces. Within that, we have a couple of projects right now running. One is on phosphorus, which is part of this big EU project, PTRAP, which we are partner of, where we look at iron 2 mineral, iron 2 phosphate mineral, vivinite dissolution, and how these redox reaction or oxidation of iron releases the phosphate, and can use that in agriculture applications. Another project we have is on copper, where we look at methanobacteria, how they get copper as a nutrient in marine system, and because it's pretty hard there because of copper sulfide, low solubility, it's hard to get the copper from there. So they use some organic ligands, and we try to mimic that in the lab environment. We also look at mineral transformation, or what kind of we have to look at when we talk about redox interfaces. So there we have interest in reductive dissolution of iron oxides, and sometimes we also do ligand promoted dissolution of iron oxide and see how re-precipitation and mineral transformation impacts the rest of the nutrient and contaminant fate in the environment. So with that little background, I will move on with my talk today, and I will focus on these 2 parts from what I talked before. Do I see my cursor? No. Okay, just give me a second. Do you see my cursor moving? Or?
No.
Oh,
now it's moving, I think.
I'll try to.
Yeah.
Okay. Better now. So today, this talk involves these 2 parts of redox interfaces. We looked at contaminant mobility and mineral transformation. And before I start the experimental part, just a background on why we look at these iron oxides and their reductive transformation or redox cycling of iron, because simply from deep hydrothermal vents all the way up to Mars and everything in between more or less has iron, and it does impact heavily our environmental processes, particularly the fate of contaminants and the nutrient in the environment. So iron redox cycling is really important for our part of research that we all enjoy. And to main focus here is that iron redox cycling has two parts. One is the reductive dissolution. So you have iron oxides, you could reductively dissolve them and release iron 2, which is also redox active. So it could impact the contaminants and nutrient, but it also can reprecipitate and the cycle continues. Other side of the story is, which I will focus on more today, is sulfidation. So you have sulfidation of iron 3 oxides, results into iron sulfide precipitation. And that is during the precipitation, you could have, in principle, contaminants trapped into the structure and precipitate. That's a solution for any remediation strategy. And for other side of the story is oxidation of these iron sulfides that could release the originally trapped contaminant in the cycle. So these both cycle continue to operate in the environment, and that's where we try to play around our research. Today, example, I will talk about ferrihydrite sulfidation. Ferrihydrite is iron 3 oxide, and how does that impact the arsenic mobility? That's the, let's say, the broader story, and then we will go in further details with that. Ferrihydrite, because it has really high surface area, approximately 300 meters square, very small particle size, 4 to 7 nanometer. And it's one of the minerals, you all are mineralogists, so you know more than me, but it's one of the mineral that stays in nanoparticulate form in the environment all the time. So when we have ferrihydrite and we let it react with dissolved sulfide, in our case, what we did, we played around with the sulfide ratio. So we provided sulfide ratio from 0.1 to 2, so oversaturating with sulfide, and try to see how this transforms the mineral and what happens to aqueous and solid phase environment. This work, however, is already been published. For the solid phase, we had this study published in 2018. And for the aqueous part of this story, exact same experiment we just published a couple of months ago in the same journal in Environment Science Nano. So that part is done already. And based on that, we were moving on that story that I'm going to talk about today, is what happens when you have contaminant sorbed on this ferrihydrite and then you react with dissolved sulfide. So all the process that we understand already, how does that impact mechanistically the fate of these contaminants? We use for the sulfidation of ferrihydrite, what we found in these already published studies is that when we change the sulfide ratio and with the same amount of ferrihydrite, what we see is that as we increase initially the sulfide ratio up to 0.5 of sulfide to iron ratio, we see the increase in aqueous iron 2 concentrations. Once we go beyond that ratio, we don't see aqueous iron 2 in the solution. So that's interesting, kind of sets up a boundary that at certain ratios, you will start to release the iron 2 and it peaks at 0.5 ratio, and beyond that, we don't see iron 2. When we look at the solid phase to confirm that, what we see here is up to the ratio of 0.5, where we see maximum of iron 2 release, we only see iron oxides in the solution. As we go beyond and we start to see less of iron 2 or no iron 2 in solution, we start to precipitate iron sulfides. So that makes sense. You have a partition, as you increase the sulfide ratio, as the stoichiometric ratio changes, you initially have reductive dissolution. So you reductively dissolve the ferrihydrite surface. So iron 2 comes into the solution. As the sulfide concentration increases, there is enough sulfide to bind with that iron 2 and precipitate as iron sulfide. So far, simple system here. And here we are interested, or we are looking at how, if in case of contaminants that are sorbed on ferrihydrite when it's exposed to sulfide, what would happen to them during this transformation or partition. And that's something we will talk more today. We have reductive dissolution of ferrihydrite. You have iron 2 release, and the second part we saw is that you changes the phase, which means that you change the surface. You have iron. You start with iron oxide, and now you are iron sulfide, and with that there's a lot of things: surface charge, surface properties, and how they interact with the contaminants. And you guys are best to know all that, so I'm not gonna go too much detail here. Just a brief introduction about how we set up this experiment. It's a very simple lab-based experiment. What we did, we took ferrihydrite, a fixed mass of ferrihydrite, and according to that, as I mentioned before, we exposed to a different concentration of sulfide. In this case, we added iron 3 or arsenic 3 or arsenic 5, two separate experiments, not together. So we did exactly same set of three set of experiments: one ferrihydrite salt with arsenic 3 on the surface, one with arsenic 5 on the surface, and of course one control. My cursor still seems to be not working, but I guess we'll work like that. So we have three set of experiments, identical. Only difference is that in one case ferrihydrite has arsenic 3 sorbed on the surface. Another case we have arsenic 5 sorbed on the surface. We followed all the aqueous parameters, the more general ones, and also solid phase parameters, looking at iron, arsenic, and sulfur speciation. Some of them I'll include today in this story. So this is simplistic experimental setup in lab, and now we move to one thing I didn't mention before. The reason for choosing arsenic is that because it's global contaminant, but I guess the last talk already might have covered that part, so maybe I will not go too much detail. One thing I would say though is that this is a map of arsenic contamination in the world, and when you when you put together this map with a newer map with the global vulnerability of sea level rise, you see that pretty much the areas, my cursor is not working, but you can look at pretty much the areas that are highly hotspot of arsenic are also most vulnerable part when we think about sea level rise, and not so a distant future in 2050, which is less than 30 years now. So not so distant future, we can see that already that part is overlapping. And so what does it mean is that when sea level rise happens, the areas that are oxic today or dry today will get submerged, so redox cycling will start, and then contaminant behavior will change, and so will be the groundwater quality. So the whole concept will impact millions and millions of lives, and especially in part of the world I come from, India has a huge population that is exposed with arsenic containing drinking water issue. So is neighboring Bangladesh. And so there are different parts in the world that are highly vulnerable to this particular combination of the story. And that's why we looked at the arsenic in this case. Coming back to our experimental setup, simplified, I think I explained already. We have three set of experiments: one control, one where ferrihydrite is with arsenic 3 on the surface, and another one arsenic 5, and we exposed with the different sulfide concentration. What we look here is the aqueous iron 2 release. You saw the figure before in the control, so it's exactly the same. As we increase the sulfide ratio up to 0.5, we see the increase in iron 2 concentration in solution, and after that we don't see. We talked about this before, so it's what we expected already. When we look at arsenic in the species, in the aqueous phase, what we found that arsenic was only released at the highest sulfide ratios of 2. What we had originally anticipated, that arsenic will be released as the reductive dissolution of ferrihydrite starts. So you start to have iron 2 in the solution. So we expected that surface soaked arsenic would also come into the solution. So we thought we would see iron 2 and arsenic at the same time, but that's not the case. What we see here is that the orange line is iron 2. So when iron 2 is in the solution, peaked at 0.5 ratio, goes down around 1, and that's when arsenic starts to come up after iron 2 is already precipitated. So that was a bit interesting because we expected the other way. But anyway, that's the result right now. What does it mean is that possibly at lower ratio, either one thing could be the arsenic is not completely, the surface is not completely saturated by arsenic. So there are some spaces where sulfide could still react, iron could still release, and arsenic could still be on the surface, because at lower ratio, we know that there is still iron oxides in the system. Another part could be that arsenic is indeed getting released, but before we detect in the solution, it could already be precipitating with different mineral phases that potentially be precipitating in the system. So if there is a new iron oxide surface available, there might be some sorption or co-precipitation there. It could be a possibility, and we'll look onto all these points as we go along. At higher ratio, the reason for release might be because we changed the surface properties. We changed from iron oxide to iron sulfide. So the surface properties are changed. So now arsenic is not, doesn't have the same affinity as it did have on arsenic oxide. So that might also be leading to release. There could be arsenic sulfide precipitation because there is iron sulfide precipitation. So there is a possibility of that. And we already know from our previous work we published in 2013 during my PhD that the sorption behavior of iron oxide is completely different than of iron sulfide when it comes to arsenic species. So we already know that part. So looking at all these different possibilities, we try to do different analysis to understand these much in greater details, and that's what I'm going to talk about next few slides. Just briefly here, you guys already know all these things about mineral changes and PGC values and what does it mean in terms of surface charge and what does it mean in terms of sorption of different contaminants. So we start with ferrihydrite. The PGC value is more or less 7.9 to 8 depending on the system you have. But when we talk about iron sulfide, that's the system is moving towards, we could have mackinawite and pyrite, more general to arsenic iron sulfide species. Mackinawite has PGC value of 2.9, so quite acidic. So, and pyrite is also in the same range, but even lower. What does it mean is that the surface charge on these different conditions will be different, so the sorption behavior will be different. So that might lead to the release of arsenic. Coming back to our release in aqueous media, we see iron 2 release, and when iron 2 starts to go down, we see the arsenic. That's what we saw in our experimental results. Question here then would be that in which form this arsenic is releasing. We had two systems. One we had arsenic 3 soaked on ferrihydrite, and another set of experiment we had arsenic 5. So the logical question was to see in which form actually the arsenic is releasing. So just to, before we go to the next slide, you all probably already know, or the last talk might have helped, arsenic toxicity, mobility, and its fate in the environment is really governed by the speciation. So if it's arsenic 3, arsenic 5, that's the most common speciation we talk about, but also more and more we are detecting even in natural environments the thioarsenic speciation, which is also highly mobile, potentially toxic. There is still I would say a lack of broader data to determine their toxicity compared to arsenic 3, which is non-carcinogen, and a lot of problem already we see. Thioarsenic species are been around. Detection has been improved, I guess. It was difficult before, but the toxicity level is still under research to see how much it impacts. But the point is that the sorption behavior and the affinity is completely different for all these speciation. And so when we talk about arsenic, we need to know exactly what speciation it is releasing into the environment, and that's how we can determine if it's how toxic it is. I mean, there's nothing good about the poison. It's toxic, less toxic, or more toxic, but we need to know how much. So for that reason, we took some samples and we collaborated with Professor Britta Planer-Friedrich in Bayreuth University in Germany. So we sent her samples, and she's one of the few labs in the world, actually, quite only 3 or 4 labs could very well determine these species in greater details that Britta does. So we collaborated with her, we sent her some samples, and she was very kind to do all the analysis. And so what we found in this system is that the arsenic
Naresh?
that we, sorry?
Naresh, sorry, I may have missed that. What is the pH of the solutions in your experiments?
pH is 5. Sorry, I probably
okay,
did not
thanks.
even say.
Okay, 5. Okay, thanks.
Yeah. we needed to know what exact speciation of arsenic is in the solution. So we did different speciation analysis, but we found this one first figure is about the system where we had arsenic 3 initially soaked on the ferrihydrite, and then it was exposed to sulfide. What we see here is that the red line is arsenic 3, which is known as arsenite. What we see here is as we increase in sulfide ratio, I don't enjoy, sorry, I can't move my cursor, so I'll try one more time. If this doesn't work, I promise I won't try again. Okay. So this arsenic 3, what was the original species that we soaked on ferrihydrite before exposing it to sulfide. During the different sulfide ratio, as we increase in the sulfide concentration, what we found is that this arsenic 3 start to decrease. But what proportionally comes up is trithioarsenate. It's a very unique species already found in a natural environment, particularly in monolakes. Quite a lot of work has been done about trithioarsenate speciation, but the mechanistic understanding of how it forms in a natural system is still lacking. So that's what we were looking at here. So what we found that as we increase the sulfide concentration, the arsenic 3 concentration decreases, and what we see is trithioarsenate. We did detect a little bit of arsenate, which is arsenic 5 due to oxidation, but that in the end of this experiment, which was 12 weeks of experimental duration, what we found is in equilibrium, we have more or less same concentration of arsenite and trithioarsenate speciation. That's from the arsenic 3 experiment. Now, we also had experiment where we soaked arsenic 5 and everything else was same. So there, what we see here is that, for arsenic 5, it's more or less similar end result, but slightly different mechanistically. What we see here is that arsenic 5, initially, as we add the sulfide, you already start to see the arsenic reduction, because sulfide is a reductant as well. So we see arsenic 5 reduced to arsenic 3, quite high concentration at the beginning. But as we increase the sulfide concentration, the path or mechanistic reaction mechanism is the same, that arsenic 3 start to decrease in concentration as we go higher in sulfide, and same species trithioarsenate comes up. So what this tells us is that in both cases, either we start with arsenic 3 or start with arsenic 5, the end speciation during the sulfidation of ferrihydrite at higher sulfide ratio is trithioarsenate and arsenite. When we check different, we also did speciation at all these sulfide ratios, and it would be confusing to show here all of that. But the behavior or pattern of arsenic partitioning was the same. As we increase the sulfide concentration, you reduce the arsenic if it was arsenic 5, and you form trithioarsenate. So that was the speciation that we were forming, and that's what we were releasing in our experiment, so to say. Okay. So arsenite and trithioarsenate were the dominant species in the end. When we use ferrihydrite that had arsenic 3 or 5 soaked on the surface. For arsenic 3, we already know it's more mobile and highly toxic, but also there are indications that trithioarsenate is slightly more mobile and toxic than arsenate itself. So what we are doing here is that we are increasing the toxicity, we are increasing the mobility of these contaminants. And that was interesting to know, a good point to know. Now coming to this is all about aqueous phase so far. So now question would be, what happened to the ferrihydrite? That is the arsenic story. So now question is, what happened to ferrihydrite minerals? Because we initially talked about there is a mineral transformation as well. Is iron mineral using iron K edge EXAS spectroscopy. What we see here is that it's all started with ferrihydrite. So in control experiment where we had no soaked on the ferrihydrite, just ferrihydrite exposed with dissolved sulfide. What we see at lower sulfide concentration is that most of the ferrihydrite is transformed to goethite. But in case of arsenic, either arsenic 3 or 5, to different degree, but with the transformation led to lepidocrosite formation, not necessarily goethite. So which was interesting, but it tells us that not only the mineral transformation impacts the speciation of contaminant and the mobility of contaminant itself also influence the mineralogy of your system. So we started with ferrihydrite. If we didn't have arsenic, we would have gotten goethite mostly. When we had arsenic soaked, the mineral transformation was lepidocrosite. So it has implication in terms of remediation, in terms of how stable your system would be, and longevity of how much you can hold with the contaminant and mobility as well. As we increase the sulfide ratio, we see the same thing. Our goethite start to transform to magnetite, which is already quite known. When you have higher iron 2 concentration in solution, the system will move toward, depending on the ratio, towards the magnetite, and you start to see somewhat pyrite as well. But in case of arsenic in the system, still have mostly the lepidocrosite as a major species. As we increase the sulfide ratio further, what we see here is that in control, most of the transformation led to formation of pyrite in the system. But when we had arsenic, either 3 or 5, the system was restricted to mackinawite, not forming pyrite. Now, I'm sure you guys already know about all these minerals, more than me at least. I never studied mineralogy. But what I want to say here is that it is assumed that the formation of pyrite is a stepwise process that starts from the formation of mackinawite and eventually transforms to pyrite using some elemental sulfur polysulfide different reaction mechanism. So it seems that the presence of arsenic restricted that transformation towards pyrite, and system was more or less stuck with the mackinawite formation. So that was the transformation of solid phase, which was interesting, and that's what we were looking for, how much impact these contaminants would have on the mineralogy. So when we look at different iron mineralogy, we also could consider that the contaminants itself could inhibit or drive in a different direction the mineral transformation. So that was from solid phase. We also did arsenic KH spectroscopy, try to find out if arsenic was co-precipitated in the structure, but kept out of it, respecting the time. But if you have questions later, we could communicate on that. What we learned from this is that ferrihydrite mineral transformation is controlled by sulfide ratio, which was new surfaces formed. So when we think about that arsenic release at the lower concentration of sulfide, it might be because there was new surface as lepidocrosite, there might be surface to still soap, so that's why we didn't see arsenic in the solution. Presence of arsenic in the system restricted the transformation of mineral toward pyrite and kept it to the mackinawite. That's what we learned from our experiment, in this case at pH 5. Take-home point here will be that microbial sulfate reduction, and I'm using the word microbial, but I only did abiotic experiment, just adding sulfide, and there is no microbiologist. I just find it complicated to deal with. So as a chemist, I would just add that and see my end data, but there's no disrespect here for microbiologists. What we saw also, the change of speciation of arsenic led to the mobility. So you have different speciation of arsenic, the mobility and toxicity changes. So it's very important to look at. And we saw finally the presence of arsenic itself impacted the mineral transformation or mineral transformation of ferrihydrite, where in control system, if there was no contaminant, system would move to pyrite, but in presence of arsenic, it only restricted to mackinawite. At this point, I would ask if I still have a few more minutes. I would like to give a quick example of field study, because so far I'm only talking about pure lab-based experiment, and it's important to see the relevance in the field. But if I don't have time, I could just stop.
You can, no, you can continue.
Okay. So that was all lab-based, pristine environment. We control most of it, so that's easy to manipulate. And we try to understand mechanistics in the simplest experiment, but nature is not as simple as we want to make it in the lab. So it was also important to see that how actually these happen in the field as well, exact these mechanisms. So what we did to look at that, and reason for that is also, and this has been questioned from the microbiology, that's why I apologize before. Usually in redox ladder, what we see is that iron reduction should happen before the sulfate reduction. So ideally, you could argue that, well, when sulfate reduction happens, probably there is no iron oxide to react with because it's already reduced. And that was the point, to see if in nature exactly that happens or not. And the answer to that in one word would be redox heterogeneity, but it's a vague term, and who knows what. To give an example. So we wanted to look at, see everything clearly in the lab, but it's a control experiment and very simplified, and there's no co-contaminant. So ionic strength is very controlled, and in nature it's more complicated than that. So what we did was, we were working at that point at Wyoming site in Riverton, focused on uranium. That was the focus of the project. We were looking at and how seasonal changes in the Colorado River water table impacts this mobility around that area. It's a historical DOE site from weapon storage facility, or probably testing facility. So most of you are aware, and some of you have worked on that, I guess, as well. So we were looking at that. Just to make it clear, we were looking here only for uranium in the beginning. And the reason I'm including that was that some arsenic in the sediment, we knew about it, but it was like 1 ppm. So traditionally, you don't worry about that. Concentration was too low. So we were only focused on uranium. But it turns out that even at lower concentration with sulfidation and redox heterogeneity, things could be more complicated. And that's the example I want you to include here. Okay. So Riverton, some of you might have visited already. There's Colorado River, there's floodplain, there's a change in the water table during the year. You get flooded, then you get dried. So you have wet, dry cyclings, and iron redox cycling keep happening. It stays for at least 2 to 3 months. The area is submerged, so all the iron is reduced, and we have taken sample at different seasons and looked at the mineralogy, and we know that a lot of iron transformation happens during this period. What we were interested in here is that during this or water table rise, what we saw is that there are some of these organic rich clay clumps, what we named or call them to make it easy, naturally reduced zones. What it is actually, there are small lenses for a few centimeters to sometime bigger or even smaller. So they are like clumps of clays, but super organically rich, and they kind of remain reducing even when the system is dry and the water table is gone. So we were suspecting these natural reduced zones are driving these redox cycling, even if it's not submerged. And that's what we wanted to look at. So we picked up some of it. It was trickier to do that because they were smaller in size. So we picked up quite a bit of those reduced lenses, that we call them, brought back to the lab and brought some sediment from the site as well. And what we designed in lab were using the same set different column experiments. So these are 12 columns, 3 control and others in triplicates with the different variable concentrations conditions. And the column also allowed us to take samples in between. My cursor is again stuck, but you can see here already that you can take samples in between during the depth of the column. So it see the transformation along the sawtooth in this system. Important to note here is the arsenic concentration is the red circle. As I said, it's pretty low, 1 ppm approximately. And so initially we were not worried about it, and we were just looking at how this transformation would happen for uranium. In this picture on the right, you can see these blackish clumps. That is the natural reduced zones that I mentioned. These are super rich in clay, super rich in organic matter, and highly reducing. So we clump them, some of them together, and we put in these columns. And the way we arranged was, this is the control column where we didn't put any of these reduced zones. And idea was to create this redox heterogeneity in the system. That's what we were looking at. So in a set of columns triplicate, we added just one of this, and in 3 columns we added 2 of them and 3 of them, just to increase the redox gradient, redox heterogeneity, to see how does it impact uranium and arsenic. I will only talk about arsenic here today. So we did follow a lot of parameters, that geochemical parameters we needed to follow, but what I want you at mainly arsenic concentration. As I said, the concentration were very low, so we were not worried. And what we found during when the sulfidation started and around this redox heterogeneous condition that we created, most of the time, this is the dash black line is WHO limit for arsenic in drinking water, which is 10 microgram per liter. And what we see here is almost all the times when we had these reducing zones introduced, all the time arsenic was much higher than that. Even at when we just added one of this, the arsenic was already higher than the allowed permissible limit. And what we also saw in the speciation of arsenic is not just the release, it's also the change in the speciation. So what we see was there's quite a bit of thioarsenic and arsenite also forming. And so it turns out that even at the low concentration, the redox heterogeneity could create that could accumulate your contaminant and increase the localized concentration that could be eventually a problem for drinking water purposes. So that kind of corroborates with the lab. And with that, I will say thank you to people who helped in this. It's quite a lot of people who did. This guy, most of you probably know, Vincent Noel from Eurolab. He was PhD here. And a lot of people you already met. Karim knows almost every one of them, so no need to introduce. And with that, I will say thank you. But before that, I will say that we are currently looking for a PhD candidate. If you know someone, please recommend, please apply. It's already open for next couple of weeks. But we have a project to see how ancient DNA interacts with environmental minerals. Idea is to see if we can find a way to find ancient DNA if we don't find the human bone or animal bone just by minerals, and can we take the useful information out of that system. So understanding interaction of DNA with minerals. So it's a 4-year PhD. It's currently available. Please apply. If you know someone, please forward, or please let people encourage to apply. And with that, finally, I'll say thank you. And if you have questions, I'm happy to be here. Don't know if I have answers, but we can talk about it. Thank you.
Thank you, Naresh. Thank you for a really nice talk. With that, I think I'm going to take questions from Karim, but before that, I just want to give a shout out that if you have questions, either you can raise your hand in the chat, or you can even also type your question in the chat. So all the question options are available. So Karim, since you raised your hand first, go for it.
I was actually applauding, and but it was an applause, I think.
Okay.
But I have questions, so I can go on if you agree.
No, that's okay.
So thanks, Naresh, and yeah, it was nice also to see these pictures of people in the end. I mean, yeah. So few questions. So yeah, initially I would have imagined that when you sulfurize the ferrihydrite, arsenic, as you were saying, would get trapped by the precipitation of pyrite, and so this is not what happens. What is happening? And so I was just so wondering, first of all, and this probably relates to the with it?
So to answer that, as we saw in controlled experiment where there was no arsenic, we saw the formation of pyrite, and when we had arsenic, it was formation of mackinawite. And what we found with exaph is that arsenic was not interactive with mackinawite, which also makes sense because it's a nanocrystalline in structure and it's unstable phase. It's not supercrystalline, and it does not have very good affinity to sorb either. And so, but what we did find, and which I did not include in this talk, and thanks for asking that, at in between concentration, let's say sulfide concentration of 0.5, for example, where we had a little bit of magnetite as well to see, there we saw some possibility, but its concentration were also low, so it's not heavily trapped there, but it's a possibility there, yeah.
And do you have any idea about what prevents or hinders the transformation of mackinawite to pyrite? Is this related to any, I don't know, passivating layers that grow on mackinawite, or?
I will give you two sides of that, and then we can together decide which one makes more sense. One thing is that it is known that the transformation of mackinawite to pyrite includes involvement of sulfur species that are elemental sulfur or polysulfide sulfur species that form in the system, and they react with mackinawite surface to create localized condition for pyrite. That's how the nucleation starts. So far, that's the understanding. So in this case, when we have arsenic, we could assume that arsenic could bind with elemental sulfur to form these arsenic species. So quite some of sulfur that would have gone to mackinawite to allow it to formation of pyrite was used by, so to say, binding with arsenic to form thioarsenic sulfate. That's a one possibility that I see. And other thing could be that because arsenic could passivate the surface, but in this case, what we saw is that release in the solution. So when the mackinawite formed, arsenic was actually in the solution, not on the surface, but might not be all of it. So it might have contributed, but I would believe that the first part of that story might be more relevant in this system.
Okay, so I probably have all the questions, but I'd like to let just go ahead now, probably.
Okay. Well, as I said, anyone who has a question can type it either in chat or just raise your hands and I'll unmute you. Well, while we're on the topic, what Karim brought up, just a quick question on thioarsenite, arsenates. I'm just wondering, so you saw the formation of that species, whether it was in the environment or in the control lab cultures. But is it susceptible to transform under redox fluctuations back into some other arsenic species? Like, could it go back into like arsenic 3, like, or get recaptured into within another iron mineral, which are, or like iron sulfur minerals present, or under oxic condition? What happens to its fate as the redox conditions fluctuate?
It's a very good question. So that's why I mentioned that the whole mechanistic story side of these species are not completely clear, but there is already some understanding to answer or give some hints to your question, I guess. So these species are not fully stable species, I would say. So they are metastable, so they keep changing in between, and that's what we see in more specifically in experiments Brita did in monolake, where as they go with the depth, the speciation changes, and the seasonal variation, you could see the changes in speciation. What it does is that level of oxidation, which you have these thio species, if I can explain in a layman term, you have arsenic and then you have oxygen around it, what is arsenate, right? And each oxygen is then substituted by the sulfur to form this thioarsenic. So as if you go to oxic system, you could potentially come back to arsenate. And if you go to more reducing system, I can imagine that if there is something to partition with sulfur, you could go back to arsenite, but it's more likely transformation during the oxidation side of things when it goes to arsenate, and that could also come back to reducing arsenite. So from arsenite to thioarsenate is less likely to revert back because there's already sulfurs around it, which kind of keeps it in the solution. And in the oxic side of things, you could substitute that sulfur again with oxygen, and then arsenate could form. That is pretty known.
Okay.
One thing I would add here is also just recently, actually, a few months ago, there's a new study from Britta about these species because she's interested in them. They measure these in Italy and in China in rice fields. So it seems that these are stable enough to stay and taken up by arsenic plants. It's pretty new. I don't know the whole details, but it seems like these species could still be interesting for environmental implication to look at because they can partition in different systems.
Okay. In that instance, if they have some stability, then it also questions if they can be of some sorts of sorbents in these conditions of like some other contaminants which may be present, and they actually have their own cycle for like what species they may interact with, especially.
Yeah.
So that would be something.
There is something, as I said before, more to be understood about these speciation because not many people are working, and challenge with that is the detection of these species. It's quite challenging because you have to have HPLC combined with IC and ICP-MS, and it has to all go in anoxic environment if you want to be more or less close. And still you see depending on the element you use in HPLC, you could still oxidize. So I'm no expert, but a discussion with Britta led me believe that maybe I wouldn't set up a lab to detect them. I would rather send the samples. So there is challenge in analyzing, and that's why there is slow progress in understanding. For the sorption part, I think I included, this is a study we published a few years ago, and we looked at different arsenic speciations, including thiospecies, to see for iron oxide and iron sulfide. What we found here is that thioarsenate species are not good, do not have good affinity for iron oxide or iron sulfide. They seem to be not so good on the sorption side of things.
Okay. Thank you. Karim, you have a question?
Yeah, just relating to that. I mean, I was kind of excited by this, the fact that you mentioned that you can find them in monolake.
Yeah.
Because that's an alkaline lake, and there are many nice carbonate rocks that are forming there. And some people have tracked the enrichment in arsenic in some of these rocks. And so I was just wondering how all these things might relate. So the thioarsenite there in monolake, is it so you form that in the sediments, right? Or
a water table, that's what was measured in Britta's work.
Okay.
I'm not sure they have done anything with the sediment. I'm not aware of. I know that the water table she measured during her work in monolake.
Okay, that's interesting. Okay.
But nowadays we have enough, so to say, good references to measure solid phase as well to detect these species if they are there.
Okay.
But we didn't do it.
Okay. This thioarsenate species, it's really exciting, I will admit. It just seems like it is. I understand it's very difficult to measure, and I appreciate when you can really detect with such a complicated methodology. But
it's still, I would say that there's no universal acceptance of this methodology either. That's the best we can do today. But the paper I showed you that we published with this data, we actually got a comment from comment that is published now for that fact that one lab in the US, let's not name, you probably all know that, the kind of challenges that the method we used was probably not the best method, which could be a case, but we thought it was pretty nice. And so there are some discrepancies there. People believe that there could be, depending on the element that you use, the question is still that, okay, you can detect what reaches to your detector, but during your process, could you oxidize already, or something could happen during your analysis? How do you detect that? So, I mean, I think they will improve as we go along, I guess. detection.
Yeah, yeah, I think it's yeah, it's difficult to. I mean, if there's that's the method, and I mean, if someone there are only few labs who are doing it, it's just difficult to answer.
It will take longer time, I guess, because Britta,
Yeah.
for example, is fully involved in just focus on these species, and she has made tremendous contribution to this field already. And I think going forward, we might have possibly easier detection of these species, I guess.
Yeah. Naresh, can you read the chat? Because there's a question.
No, I cannot actually.
Okay. So I'm gonna.
Maybe maybe I can.
Okay.
Yes.
There's
yes.
a question from Elodie.
Question from Elodie. Do you know why you are not forming arsenopyrite in your system? Only iron sulfide, but is needed to form arsenic sulfide. As we said already in the result, that when there was arsenic in the system, pyrite did not form. So the question of arsenopyrite is not there. Why they are not forming? As we, I think Karim asked that question already, and it's related to that the system is restricted toward mackinawite and not moving to pyrite. If it was the case, and arsenic we didn't detect in the solution, we could have assumed that there was arsenopyrite, but in this case it wasn't.
So you need sulfur, elemental sulfur or polysulfide to form arsenopyrite also,
well,
not only
I would say
pyrite.
to make pyrite, and arsenopyrite is pyrite that has arsenic.
Yeah, because I didn't know, because for arsenopyrite it's one arsenic, one sulfur, and one iron. So I was wondering if, based on ferrihydrite or mackinawite, you can just add arsenic to the system to, I don't know, precipitate directly, but it's not working. So
in this case, it wasn't precipitating as pyrite. Yeah.
Okay, thank you.
But I would also add here that the pyrite story is, even though old, we see papers from I don't know 70s, and Ricard and Luther did a lot of work on how iron sulfide formation happens, but people still question if that's the only pathway to form pyrite from mackinawite, to having the elemental sulfur and polysulfide. But I don't know what else pathways are, but there seems to be some gap. People believe that there could be other pathways, but I don't have that answer.
Karim?
Yeah, so I guess Arnaud Duverger, who is around, might have some ideas about that. But I was also wondering, so on the same line, so you chose this pH of 5, and I was wondering why 5, and would you expect things to go differently if you were like, I don't know, a much higher pH or a little lower pH? I have no clue about what are the constant reaction constants for the formation of these thioarsenite species, and the solubilities of the different phases we are talking about.
So one thing to answer first is the pH thing. Yes, we know for a fact that system will move slightly differently. And the reason we know it is that we did quite few of these experiments. Some of them are published, some aren't. We also did at pH 5, not with arsenic, but in general, the sulfidation. We saw the difference between 5, and especially in formation of pyrite. So at pH 5 seems like pyrite is more likely to form in control experiments, and if you move pH towards 7 or above, there are some constraints there. And for arsenic speciation, there's no known enough data for constants for thioarsenic species, but it does make a difference for arsenate and arsenite. Yes, so it might have been different at pH 7. Yes. And we are actually doing that now at different pH, but I don't have the data to show right now. And the reason that we all write papers and not say clearly, in this case, the reason to do at 5 was that because we knew in control we will have pyrite, and we wanted to see if that would play along.
I guess if no one has a question. I'm going to ask one quick question.
Sure.
So I know you said you want to stay away from microbiology, or at least you did the abiotics experiment. But in your opinion, I suppose you spoke briefly about in the end about the environmental. But if there were microbial species involved, do you think there would be the inhibition of mackinawite to pyrite formation would no longer, like you would be able to observe pyrite in your system with arsenic if there was microbial species present?
I can't say for a certainty that it wouldn't, so it's a possibility. What I can say is that the transformation still depends, or so to say, controlled by the ratio of sulfur that is available to the iron. And the reason for doing, with all due respect with microbiology, the reason for doing that in abiotic is that you can control that ratio. In biotic experiment, we don't know how much bacteria we can measure, but we will never exactly know. So the reason to do it abiotic was that we could control these exact ratios that we want to provide. It's possible that with the, because you also not just about the sulfide coming for bacteria or abiotically, during bacterial activities, the redox parameters also slightly localized change, so that could contribute to slightly different results.
Right, exactly. I mean, yeah, sure. I mean, abiotic system definitely it has an advantage to see all these effects of these parameters in a, and you can pinpoint exactly what's going on. I just thought it would be really cool to also just add microbes and just see.
Yeah, to add to this answer, the study I showed from the field, there we also did arsenic iron speciation of these columns, and we saw that most of arsenic was mackinawite, and that's where we see the arseno or thioarsenic species. So it seems to corroborate with the how it works in the field. There we didn't control it; was just microbial system. There were natural microbes that we drive on the sulfidation. Yeah.
Okay, pretty cool. Okay, so I guess we are just in time, and I don't see other questions raised, neither on chat. So with that, I would like to thank Naresh for a really good talk, and thanks for taking time to share your results with us. And as I can speak on behalf of everyone, it was really informative and very useful just to think about all the arsenic and thioarsenates and learn about it. Thank you so much.
Thank you so much for inviting, and I really enjoyed the discussion. And I haven't seen Karim since Goldschmidt 3 years ago, I guess, and so it's good to see everyone here as well, and everyone else will have a chance to meet, I guess, at some point at Goldschmidt's, I guess.